The ophthalmic artery is derived from the internal carotid artery. It gives rise to several branches that divide it into two main groups. The orbital branches, which are colored orange here, supply the structures of the orbit, the eyelids, and the lacrimal gland. And the ocular branches, which are colored dark red here, supply the eyeball itself. Those branches are remembered by the mnemonic Dr. Macula, but not in anatomical order of branching. Those branching in the typical order are: the central retinal artery is the first branch of the ophthalmic artery, and it is ocular branch. Lacrimal artery supplies the lacrimal gland. Posterior ciliary arteries, short and long branches. Muscular branches supply extraocular muscles. Supraorbital artery supplies upper eyelid and forehead. Posterior and anterior ethmoidal arteries supply ethmoid sinuses and nasal cavity. Internal palpebral arteries supply eyelids. Supratrochlear artery also supplies the forehead. Dorsal nasal artery. These orbital branches will be studied in more detail later. What is important now are the ocular branches of the ophthalmic artery. The central retinal artery is the first ocular branch of the ophthalmic artery. It arises just after the ophthalmic artery enters the orbital cavity through the optic canal, inferior and lateral to the optic nerve. From its origin, the CRA runs forward and medially, then ascends to approach the inferior surface of the optic nerve. At a point approximately 10 millimeters behind the eyeball, it pierces the optic nerve sheath, specifically the dura mater and arachnoid mater surrounding the nerve, and enters the central core of the nerve. It continues this intraneural course until it reaches the lamina cribrosa of the sclera. At the level of the optic disc, the CRA emerges from the optic nerve head and immediately branches into four major retinal arterioles: superior temporal, inferior temporal, superior nasal, inferior nasal. These branches course along the nerve fiber layer and form two major arcades in both the superior and inferior hemispheres of the retina. They supply the inner two-thirds of the retina, from the nerve fiber layer to the inner nuclear layer. The CRA is an end artery. It lacks significant anastomoses and collateral circulation. As a result, occlusion leads to ischemia and results in sudden, profound, painless monocular vision loss. The second major ocular branch is the group of the long and short posterior ciliary arteries. The short posterior ciliary arteries are usually 6 to 12 in number. They pierce the sclera around the optic nerve. They form the choroidal circulation, supplying the outer third of the retina and the optic nerve. A branch of the choroidal circulation, often one of the short posterior ciliary arteries, may give rise to a ciliary retinal artery found in 25% of individuals, supplying part of the macular region. Importantly, unlike the central retinal artery, these arteries have anastomoses and partial collateral circulation. These two ocular branches, the central retinal artery and the posterior ciliary arteries, form the main vascular supply of the eye. The venous drainage of the eye mirrors the arterial system in many ways. There are two major venous systems: the central retinal vein draining the inner retina, and the vortex veins and ciliary veins draining the choroid, ciliary body, and iris. The central retinal vein runs alongside the central retinal artery. It begins at the optic disc, formed by the confluence of four retinal venules, which correspond to the arteriolar arcades. It drains the inner layers of the retina, from the nerve fiber layer to the inner nuclear layer. From the optic disc, it follows a path posteriorly through the optic nerve within its central core, just like the central retinal artery. As it exits the optic nerve, it pierces the meninges around 8 to 10 millimeters behind the globe. The central retinal vein has more variability in its drainage pattern. In most cases, it drains directly into the superior ophthalmic vein, but in about 20 to 40% of people, it drains directly into the cavernous sinus. The vortex veins are the main drainage system of the choroid. There are typically 4 to 6 vortex veins located in the equatorial region of the globe, with one vortex vein, at least in each quadrant. Each vortex vein drains a quadrant of the choroid, ciliary body, and peripheral retina. They exit the eye by piercing the sclera obliquely posterior to the equator. The superior vortex veins drain into the superior ophthalmic vein. The inferior vortex veins drain into the inferior ophthalmic vein. Importantly, these veins are valveless, allowing retrograde flow, which is why infections from the face or orbit can potentially spread to the brain through the cavernous sinus. The choroid lies between the retina and the sclera. It is thicker posteriorly than anteriorly and consists of 5 layers from outward to inward. The outer avascular layer, called suprachoroid lamina, consists of pigmented connective tissue. Then the choroidal stroma, which consists of the outer Haller's layer, contains large vessels, and the inner Sattler's layer contains intermediate vessels. Then the choriocapillaris layer contains capillaries with fenestrated endothelium surrounded by a basement membrane and parasites. Those can be seen on OCT beneath the innermost layer of the choroid, which is Bruch's membrane. Bruch's membrane is a pentalaminar structure consisting of 5 layers from outward to inward: the basement membrane of the choriocapillaris, an outer collagenous layer, then an elastic layer, then an inner collagenous layer, and finally the basement membrane of the retinal pigment epithelium layer. Between the choroid and the retina, there is an outer blood retinal barrier, which forms from Bruch's membrane and the tight junction between the RPE cells. This barrier plays an important role preventing leakage of fluid from the choroid to the retina. Remember that all the choroidal layers are supplied by the choriocapillaris, which receives blood from the short posterior ciliary arteries. The retina consists of 10 layers by the organization of its cells. The retinal pigment epithelium cells, the photoreceptor cells, are the rods and cones, which consist of outer segments towards the RPE cells and inner segments, then the cell's nucleus. Those cells synapse with bipolar cells that also synapse with ganglion cells. Ganglion cells' axons form nerve fibers that merge into the optic nerve. Other important supportive cells between the bipolar cells are the horizontal and amacrine cells. Muller cells are supportive cells, and unlike other cells, they extend vertically, forming the outer limiting membrane in the site where photoreceptors pierce those cells, and the inner limiting membrane that sticks with the cortical vitrea. All those cells form 10 layers organized from outward to inward: the retinal pigment epithelium layer, then the segments of the photoreceptors, then the outer limiting membrane, followed by an outer nuclear layer consists of photoreceptors nucleus, then an outer plexiform layer between the photoreceptors and bipolar cells. An inner nuclear layer is formed by the nucleus of bipolar cells. This layer also has the horizontal and amacrine cells. Then we have an inner plexiform layer between bipolar cells and ganglion cells, which form the ganglion cell layer. The axons of those cells form the nerve fiber layer that gather in the optic nerve. The inner limiting membrane formed by the basement membrane of the Muller cells. The outer third of those layers is supplied by the posterior ciliary arteries through the distribution of nutrients from the choriocapillaris, while the inner two-thirds of those layers is supplied by the central retinal artery through two capillary plexus. The superficial capillary plexus, located in the nerve fiber and ganglion cell layers, and the deep capillary plexus, located in the inner nuclear layer. Those capillaries consist of only one layer of endothelial cells, lying on the basement membrane. Tight junctions linking endothelial cells form the inner blood retinal barrier. Pericytes, with their processes, are located around the endothelial cells and have contractile properties. The retina is divided into several regions. The macula is the center 5.5 mm of the retina. The perimacular region surrounds the macula and lies just outside the vascular arcades. Around the equator is the equatorial retina. Anterior to the equatorial retina is called the peripheral retina, which ends at the ora serrata. The ora serrata extends to the pars plana of the ciliary body through jetties of retinal tissue called dentate processes. Pars plana also extends to the ora serrata forming ora bays, which may be closed by connection of two adjacent dentate processes to form enclosed ora bay. Thickened retinal tissue extends into pars plana called meridional fold, which may be aligned with a ciliary process to form a meridional complex. Such meridional complexes can cause traction on the adjacent retina and can lead to formation of a retinal tear. Most retinal tears, anyway, occur in the peripheral retina anterior to the equatorial retina. The macula is also divided into several regions. The central 1.5 mm of the macula is the fovea, which is equal to the optic disc diameter, and photoreceptors are entirely cones. This region is responsible for color vision and high acuity. The half-millimeter ring around the fovea is the parafoveal region, where the ganglion cell layer, the inner nuclear layer, and the outer plexiform layer are thickest. Surrounding the parafovea is the perifovea, whose margin corresponds to the macula margins. The center 0.35 mm of the fovea is the foveola, which has concentrated, elongated, and densely packed cones. The inner nuclear layer and the ganglion cells layer above the cones in the foveola are laterally displaced. The center 200 micrometers of the foveola is the umbo, which is a small depression representing the fixation light reflex. The fovea avascular zone, or FAZ, is a very important landmark, particularly in fluorescence angiography. This region is about 600 micrometers at the center of the fovea, usually larger than the foveola, and is devoid of blood vessels. Unlike other retinal regions, which are supplied by the central retinal artery, the fovea is supplied by the choriocapillaris.